X-Ray of Life, Volume 1 is the first of a trilogy that outlines the foundational challenges of life's emergence, offering a comprehensive analysis of the problems involved in the transition from simple chemical compounds to the first living cells. In 1988, Professor Dr. Klaus Doss wrote, "More than 30 years of experimentation on the origin of life in the fields of chemical and molecular evolution have led to a better perception of the immensity of the problem of the origin of life on Earth rather than to its solution. At present all discussions on principal theories and experiments in the field either end in stalemate or in a confession of ignorance. The truth is, that even today, after 70 years of intensive scientific research, science has still no clue how life began on Earth." George Whitesides is an American chemist and professor of chemistry at Harvard University. In an interview in 2012, he said, "There's an intellectual problem, which is the origin of life. The origin of life has the characteristic that there's something in there as a chemist, which I just don't understand. I don't understand how you go from a system that's random chemicals to something that becomes, in a sense, a Darwinian set of reactions that are getting more complicated spontaneously. I just don't understand how that works. So that's a scientific problem." Let's see what Eugene Kounin has to say about this fundamental question of the origin of life. He is a prominent Russian-American biologist and a recognized expert in the fields of evolutionary biology and bioinformatics. Let's see what he has to say in regards of the status quo in the field of origin of life research. He writes in his book, The Logic of Chance, despite many interesting results to its credit, when judged by the straightforward criterion of reaching, or even approaching, the ultimate goal, the origin of life field is a failure. We still do not have even a plausible coherent model, let alone a validated scenario, for the emergence of life on Earth. A succession of exceedingly unlikely steps is essential for the origin of life, from the synthesis and accumulation of nucleotides to the origin of translation, through the multiplication of probabilities. These make the final outcome seem almost like a miracle. It is fascinating to see how the scientific understanding of the fundamental units of life, living cells, has progressed and evolved over time. In the 19th century, knowledge in this area was still quite rudimentary. In a letter to his friend J.D. Hooker, Darwin wrote in 1871, "But if, and oh what a big if, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity and C present, that a protein compound was chemically formed, ready to undergo still more complex changes." For example, Ernst Haeckel, a famous German biologist, wrote an article that was published in Nature in 1871. There, he wrote, "The simplest independent organisms which we know, and which moreover can be conceived, the Monera, consist in fact while living of nothing else but the simplest sided, a structureless bit of protoplasm, and since they exhibit all forms of vital activity, nutrition, reproduction, irritability, movement, these vital activities are here reduced to one of structureless protoplasm. Today, we know better. Aquifex eolicus, one of the simplest thermophilic free-living bacteria, has a genome of 1.55 million base pairs encoding 1,512 proteins. It thrives in extreme environments. Its complexity includes a sophisticated cell membrane for ion and nutrient transport, a DNA replication system with error correction, precise ribosomal translation, de novo synthesis of essential biomolecules, and ATP synthase for energy production via proton gradients. 1.55 million base pairs would fill approximately 1,000 A4 pages of a book if each base pair was of the size of a normal letter, providing the instructions to make 12 different types of proteins, with an average size of about 342 amino acids. Not only are there about 1,500 proteins types, but a prokaryotic cell contains approximately 40,000 to 50,000 total proteins per cell. And it replicates that number between 1 and 3 hours. This self-replication process is an engineering marvel. The cell must accurately replicate itself while maintaining energy production, waste removal, and other vital processes. The precision and efficiency of this system are unparalleled in human engineering. For example, if a human factory were to replicate itself, it would require not only duplicating every machine and tool but also ensuring that every worker knew exactly what to do, without errors, in a matter of hours. Four major classes of organic molecules are found in living cells. All forms of life have organic molecules and macromolecules that fall into these four broad categories, based on their chemical and biological properties: carbohydrates, lipids, proteins, and nucleic acids. Let's first give a closer look at the problems for the prebiotic synthesis of RNA and DNA. The origin of RNA and DNA would have faced major prebiotic hurdles right from the start, starting with inefficient carbon and nitrogen fixation, limited phosphorus availability, and low yields of key precursors like ribose and nucleobases. Additionally, destructive energy sources and inconsistent environmental conditions further would have hindered the formation of nucleotides precursors, essential for the formation of RNA, DNA. James Watson at Left and Francis Crick discovered the structure of the DNA, deoxyribonucleic acid, molecule in 1953. DNA are the molecules which make up the alphabet, which specifies biological heredity. DNA are the molecules which store the blueprint of life, and as such, hold a central indispensable position. The RNA polymerase machine complex transcribes the instructional information stored in DNA into RNA. RNA is built of, almost, the same four-letter alphabet as DNA. It is more fragile, and as such, it could also be an information carrier, but less adequate long-term. Who wants to find answers about how life started, needs to find compelling explanations about how RNA and DNA first emerged on Earth. In all known living beings, genetic information flows from DNA to RNA to proteins. Their work on the structure of DNA was performed with some access to the X-ray crystallography of Maurice Wilkins and Rosalind Franklin at King's College London. Combining all of this work led to the deduction that DNA exists as a double helix. This information was critical for their further progress. They obtained this information as part of a report by Franklin to the Medical Research Council. The report was by no means secret, but it put the critical data on the parameters of the helix, base spacing, helical repeat, number of units per turn of the helix, and diameter of the helix, in the hands of two who had contributed none of those data. With this information, they could begin to build realistic models. The big problem was where to put the purine and pyrimidine bases. Details of the diffraction pattern indicated two strands, and indicated that the relatively massive phosphate ribose backbones must be on the outside, leaving the bases in the center of the double helix. Details of the diffraction pattern indicated two strands, and indicated that the relatively massive phosphate ribose backbones must be on the outside, leaving the bases in the center of the double helix. Crick, Watson, and Wilkins were awarded the 1962 Nobel Prize in Physiology or Medicine specifically for their discovery of the double helix structure of DNA. Franklin having died of cancer in 1958. Nucleotides are essential to cellular metabolism, and nucleic acids are the molecules of genetic information storage and expression. Nucleotides are building blocks for DNA and RNA. The two classes of nucleic acids are deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. DNA molecules store genetic information coded in the sequence of their building blocks. DNA can be considered as a modified form of RNA since the ribose sugar in RNA is transformed into deoxyribose in DNA at the 5' positions, see the red circle in the picture above, and the uracil base is methylated into thymidine, more about this, later. The structural difference between these sugars is that ribonucleic acid contains a hydroxyl, OH, group, whereas deoxyribonucleic acid contains only a hydrogen atom in the place of this hydroxyl group. Nucleotides are the building blocks of DNA and RNA, consisting of three parts: a phosphate, a sugar, ribose or deoxyribose, and a nitrogenous base. The type of sugar determines whether it's a deoxyribonucleotide, DNA, or a ribonucleotide, RNA. The attachment sites of the base and phosphate to the sugar are essential for function, and how prebiotic processes achieve this precise configuration remains an unsolved mystery. Nucleotide bases appear in two forms, a single-ring nitrogenous base, called a pyrimidine, and a double-ringed base, called a purine. No known prebiotic route produces concentrated high-energy precursors for purines and pyrimidines. Cytosine is unstable, not found in spark discharge experiments or meteorites, and degrades easily. Guanine is hard to produce. Other RNA bases form from simple precursors, but guanine does not. Adenine requires unrealistic hydrogen cyanide levels, deaminates quickly, and has weak, nonspecific interactions. Uracil degrades rapidly. Nucleobases must form faster than they break down to accumulate prebiotically. DNA has the ribose sugar in RNA transformed into deoxyribose in DNA at the 2' positions. A base is attached to the 1' carbon atom, and a phosphate group is attached at the 5' positions. Compared with ribose, deoxyribose lacks a single oxygen atom at the 2' positions. The prefix deoxy, meaning without oxygen, refers to this missing atom. Pentose sugars are 5-carbon monosaccharides, with aldopentoses like ribose and deoxyribose found in nucleotides. DNA forms when bases link via hydrogen bonds, and phosphates connect to sugars via phosphodiester bonds. Ribose, a 5-carbon sugar, exists in 6 forms, with beta-D-furanose being key in RNA. The Formose reaction is the best-studied prebiotic ribose synthesis pathway. However, the Formose reaction is complex, requires specific catalysts, and produces ribose only as an intermediate among many other sugars, making prebiotic ribose synthesis inefficient. The Formose reaction produces a chaotic mix of sugars, with ribose forming in less than 1% yield among many isomers. Its instability prevents accumulation, requiring rapid conversion to ribonucleosides before degradation. Ribosa's instability in water and short half-life at neutral pH make its prebiotic availability unlikely. High formaldehyde concentrations are needed, but formaldehyde reacts readily in water. The ribofuranose backbone in DNA/RNA either arose by chance or was chosen by intelligence. Given the reaction's lack of selectivity and ribosa's instability, chance seems implausible. Leslie Orgel concluded that some progress had been made in the search for an efficient and specific prebiotic synthesis of ribose and its phosphates. However, in every scenario, there are still a number of obstacles to the completion of a synthesis that yield significant amounts of sufficiently pure ribose in a form that could readily be incorporated into nucleotides. There have been a wide variety of attempts and proposals to try to solve the riddle, but up to date, without success. The article in Science magazine from 2016 admits, ribose is the central molecular subunit in RNA, but the prebiotic origin of ribose remains unknown. And a recent research paper from 2018 reports, even if some progress has been made to understand the ribose formation under prebiotic conditions, each suggested route presents obstacles, limiting ribose yield and purity necessary to form nucleotides. A selective pathway has yet to be elucidated. Phosphorus is essential for DNA/RNA, forming stable backbones via phosphodiester bonds, where phosphate links two sugars. However, its poor solubility in water and terrestrial environments makes its prebiotic availability and incorporation into early molecules a significant challenge. Phosphodiester bonds form the backbone of DNA/RNA, linking the 3' and 5' carbons of sugars via strong covalent bonds. However, on prebiotic Earth, activating phosphate for this energy-intensive reaction would have been impossible, and phosphate concentrations were too low. No known geochemical process produces polyphosphates in high yields. Additionally, nucleotides require precise alignment of phosphate, ribose, and nitrogenous bases in size, shape, and bonding strength to form DNA's spiral structure. For a self-replicating RNA molecule, high concentrations of all 4 bases in one place would have been necessary, a highly improbable scenario. The prebiotic synthesis of nucleotides, adenine, guanine, cytosine, uracil, thymine, and their transition to enzymatic biosynthesis faces major challenges, selecting the correct components, right-handed configurations, ribose, complementary bases, forming precise bonds, glycosidic, phosphodiester, hydrogen, and overcoming instability, energy requirements, and water's destructive effects. Additionally, achieving sufficient nucleotide concentrations and transitioning from random prebiotic reactions to modern, interdependent metabolic networks is highly implausible without intelligent guidance. These hurdles make the spontaneous origin of RNA and life's complex biochemistry seem virtually impossible. DNA and RNA exhibit a deep chemical bias, using only right-handed molecules, which are essential for proper intermolecular interactions and biological function. The right-handed double helix structure arises from the hydrophilic phosphate/ribose backbone, hydrophobic base pairing, and precise geometry, while mixtures of left and right-handed sugars create unstable, non-functional structures incapable of replication or catalysis. Biological systems exclusively use D-ribose, right-handed, but abiotic experiments produce equal amounts of left and right-handed ribose. No known symmetry breaking process explains how right-handed RNA emerged from a mixed pool of molecules, making the origin of this bias a mystery. Gerald Joyce wrote a science paper which was published in Nature magazine, in 1984, suggested that in order for life to emerge, something first had to crack the symmetry between left-handed and right-handed molecules, an event biochemists call breaking the mirror. Since then, scientists have largely focused their search for the origin of life's handedness in the prebiotic worlds of physics and chemistry, not biology, but with no success. So what is the cop-out? Pure chance. Luck did the job. That is the only thinkable explanation. How could that be a satisfying answer in face of the immense odds? But then, the same author, Christian de Duve, Nobel Prize winner in physiology or medicine, dismisses instant creation as heuristically sterile. A sterile discovery? In other words, a discovery lacking evidence. In his following book, Genetics of Original Sin, he then extended a bit further, and exposed what he meant by heuristically sterile. It is conceivable, as proposed by Nobel Prize-winning biologist Christian de Duve, that early molecules were short enough for nearly all possible sequences to exist and be subjected to natural selection. In this view, natural selection could have acted on these sequences, gradually refining them into functional biological systems. However, de Duve's dismissal of intelligent design in favor of natural selection lacks empirical evidence and relies heavily on speculation. RNA and DNA use a 5-membered ribose ring backbone, which is uniquely suited for efficient Watson-Crick base pairing. 6-membered ring structures with 6 carbon sugars, though equally likely to form under prebiotic conditions, are too bulky to support proper base pairing and cannot function as genetic systems. In contrast, nucleic acids based on 5 carbon sugars, including ribose, form highly efficient informational systems, highlighting the precise molecular design required for life. How would natural, unguided events on early Earth select what works? The author's conclusion states that optimization, not maximization, of base pairing strength determined RNA selection. Yet, why would unguided processes select molecules with no inherent function? Furanose or pyranose rings would simply disintegrate without purpose. The use of evolutionary jargon sidesteps these questions, revealing the fragility of the naturalistic paradigm. Once all the necessary components were available, they would need to be transported to the same assembly site and separated from non-functional molecules. Assembling the 3 parts involves 2 challenging reactions, forming a glycosidic bond with the correct stereochemistry to link the nucleobase and ribose, followed by phosphorylating the resulting nucleoside. The prebiotic synthesis of RNA required ribose and nucleobases to form nucleosides, linking the correct nitrogen of the base to the correct carbon of the sugar. This process, particularly for pyrimidines, remains unresolved without enzymes. Random, non-guided reactions struggle to achieve the precise, repetitive coupling needed. Additionally, activating nucleosides for polymerization into RNA poses further challenges. The emergence of catalytic polymers and the mechanisms for their assembly on prebiotic Earth remain unanswered questions in origin of life research. No known methods enable the thermodynamically unfavorable reaction of forming nucleosides in aqueous solution. While dry-phase synthesis has produced purine nucleosides, it fails for pyrimidine bases and ribose. Laboratory syntheses of ribonucleotides rely on protecting groups to overcome thermodynamic and kinetic barriers, requiring controlled manipulation of sugars and nucleobases. For a molecule to function as a self-replicator, it must be a homopolymer with a backbone composed of identical, repetitive units. In a prebiotic world, what purpose would the formation of such a homopolymer serve? Given that only random, unguided events could drive its generation, the likelihood of this happening is astronomically low, bordering on impossible. The phosphodiester bonds in RNA are highly specific, forming exclusively between the 5' of one ribose and the 3' of another, rather than the 2'. This 5' to 3 linkage is crucial, as it provides far greater stability to RNA molecules compared to 5' to 2 bonds. However, there is no explanation for how this specific bonding pattern could have been consistently selected abiotically to form functional polynucleotide chains. While nucleotides can polymerize through condensation reactions, the precise and repeated formation of 5' to 3 linkages in prebiotic conditions remains a mystery. This specificity is essential for the stability and functionality of RNA and DNA. In a research paper from 2010, John D. Sutherland reported, under plausible prebiotic conditions, condensation of nucleobases with ribose to give beta-ribonucleosides is fraught with difficulties. The reaction with purine nucleobases is low-yielding and the reaction with the canonical pyrimidine nucleobases does not work at all. Fitting the new synthesis to a plausible geochemical scenario is a remaining challenge. Origin of life research struggles to explain how monomer ribonucleotides transition to polynucleotides. Phosphodiester bonds, essential for nucleic acids, link the 3' carbon of one sugar to the 5' carbon of another. In modern cells, this bond forms when nucleotide tri- or diphosphates break apart, releasing energy for enzyme-catalyzed reactions. However, prebiotic conditions lacked enzymes and energy-rich phosphorus compounds. The main challenge is the thermodynamically unfavorable removal of water in aqueous environments, which modern life overcomes via enzymatic catalysis and pyrophosphate hydrolysis. Prebiotic synthesis would also face the issue of unordered, disparate molecules needing separation and organization. Hydrogen bond-base pairing is crucial for DNA stability, enabling its function as a repository of codified instructional information through specific sequences. The fine-tuning required for life extends beyond cosmic and physical conditions to biochemistry itself. The precise strength of chemical bonds, such as the carbon-oxygen double bond being 30 kilocalories per mole stronger than carbon-carbon or carbon-nitrogen bonds, is essential for the universal genetic code. Without this exact bond energy, Watson-Crick base pairing, and thus transcription, translation, and life as we know it, would not exist. This biochemical fine-tuning illustrates the delicate balance necessary for life's emergence and sustainability. The prebiotic synthesis of RNA and DNA faces significant challenges due to their chemical instability. RNA is particularly unstable because of the 2' hydroxyl group, which makes the strand prone to cleavage through intramolecular reactions. DNA, lacking this 2' hydroxyl group, avoids such cleavage but still presents other prebiotic synthesis hurdles. These instability issues, along with degradation and the asphalt problem, make the abiotic forma. Prebiotic processes tend to produce chaotic, useless mixtures, asphalt problem, not structured polymers. RNA degrades quickly, with no mechanism to remove byproducts, leading to contamination. Natural reactions overwhelmingly generate non-functional molecules, making abiogenesis unlikely. Stanley L. Miller noted ribose's instability, ruling it out as a prebiotic genetic material. The book, Biological Science admits, the production of nucleotides remains a serious challenge for the theory of chemical evolution. At this time, experiments that attempt to simulate early Earth environments have yet to synthesize complete nucleotides. The inevitable conclusion of this survey of nucleotide synthesis is that there is at present no convincing, prebiotic total synthesis of any of the nucleotides. Many individual steps that might have contributed to the formation of nucleotides on the primitive Earth have been demonstrated, but few of the reactions give high yields of products, and those that do tend to produce complex mixtures of products. Steve Benner is the founder and president of the Westheimer Corporation, a private research organization, and a prior Harvard University professor. He is one of the world's leading authorities on abiogenesis. This is his evaluation of what he has observed. We are now 60 years into the modern era of prebiotic chemistry. That era has produced tens of thousands of papers attempting to define processes by which molecules that look like biology might arise from molecules that do not look like biology. For the most part, these papers report success in the sense that those papers define the term. And yet, the problem remains unsolved. Stephen Benner has been remarkably courageous by admitting openly and categorically, the origins problem cannot be solved. Long periods of time do not make life inevitable. Molecules rather disintegrate based on the second law of thermodynamics. And randomization turns more complete. Since prebiotic processes are natural randomizers and abiogenesis requires specific products, it does not appear that prebiotic processes have inherent capability to meet the requirements necessitated for successful abiogenesis. This plausibly characterizes every hypothetical step of abiogenesis and explains why none have succeeded. Claude Shannon showed that randomization is the fundamental behavior and entropy is simply a mathematical expression of certain of its aspects. In 2017, Sutherland formed nucleotides, amongst all of the basic building blocks, lipids for compartments, amino acids for metabolism, starting with cyanide as a common initial substrate. However, this ended up requiring 6 separate ponds with their own unique geochemical conditions and whose products then needed to be mixed together in a specific sequence. Even this degree of complexity did not supply required products, but only precursors. The requirement of so many unique ponds and associated chemistries in such close proximity to each other stretches plausibility. And Graham Cairns Smith noted, I think, to say that on average the 14 hurdles that it would take to make primed nucleotides would each take 10 unit operations, that at least 140 little events would have to be appropriately sequenced. Unguided, the appropriate thing happened at each point on one occasion in 6. The odds against a successful unguided synthesis of a batch of primed nucleotide on the primitive Earth would be a huge number, represented approximately by a 1 followed by 109 zeros, 10 to the power of 109. How did nature start to play this game? At the very least a maintained supply of primed nucleotides would be required for any kind of organism using our kind of message tapes. A nucleotide-making factory would be needed. The odds are enormous against its being coincidence. No figures could express them. Now, let's move to the second main topic of this video, which is about the problems to synthesize prebiotically the 20 amino acids used in life. Scientists now recognize 22 amino acids as the building blocks of proteins, the 20 common ones and 2 additional, less frequently used ones. According to the scientific narrative, the hypothesis is that amino acids emerged on early Earth, between 3.5 and 4 billion years ago, through chemical evolution by means of chemical synthesis, or rather, fortunate chemical accidents. Scientists hypothesize that during the Hadean Eon, the period when life originated, there were over 500 naturally occurring amino acids on Earth. And here lies one of the major scientific problems. On the prebiotic Earth, there was a chaotic, disordered abundance of molecules of all kinds, and the vast majority of them were irrelevant to the origin of life. Yet, there was no evolutionary natural selection before life existed. In the scientific article, The Role of Natural Selection in the Origin of Life, the author writes in the conclusion, how successful are the theories, RNA first, RNA later, metabolism first, and preparatory metabolism, in elucidating the origin of life as an evolutionary process. So far, none of these paradigms can claim decisive experimental support. This means that the process that selected and organized the few molecules relevant to life cannot be explained by the mechanisms of biological evolution, as those only became effective after life emerged. This poses a significant challenge for science, as it must explain how the complex, life-sustaining structures arose from this chemical, primordial soup, without the driving force of natural selection. It is a mystery how the ordered complexity of life could emerge from such molecular chaos. In 2008, a team of researchers led by Jeffrey Botta, a former student of Stanley Miller, reanalyzed the archived samples from the original Miller-Urey experiments. Using advanced analytical techniques that were far more sensitive than those available in the 1950s, they identified a total of 22 amino acids and 5 amines in the samples. However, the reanalysis also revealed a critical limitation: 8 of the proteinogenic amino acids, those essential for life, were never produced in any of the Miller-Urey experiments. These 8 amino acids were absent not only in the experimental results but also do not occur naturally on Earth. This finding underscored the complexity of prebiotic chemistry and raised important questions about the sufficiency of such experiments in explaining the origin of life's building blocks. The next challenge is concentrating the amino acids used in life to a location where polymerization could begin. Polymer formation in aqueous environments would likely have been necessary on early Earth, as water would have been the reservoir for the amino acid precursors required for protein synthesis. However, the desired amino acids would have been extremely diluted in the ocean, with no natural mechanism to concentrate them in one place to initiate reactions. Even if this hurdle were overcome, another problem arises. Careful experiments in aqueous solutions with very high amino acid concentrations show that significant polymerization, producing proteins, is impossible in such an environment. But it gets even worse. Amino acids tend to break apart in water, not come together. Even if there were billions of simultaneous trials as billions of building block molecules interacted in the oceans or along the thousands of kilometers of coastlines that could provide catalytic surfaces or templates, even if, as claimed, there was no oxygen on the prebiotic Earth, there would still be no protection from ultraviolet light, which would destroy and disintegrate prebiotic organic compounds. The next challenge is understanding why life uses exactly 20, or in some cases, 22, amino acids, no more, no less. A related question is, why are these specific amino acids used in life, rather than any other possible set? This raises deeper questions about the chemical and functional constraints that may have guided the selection of these particular building blocks for biological systems. Why 20 and not 10 or 30? And why those 20 in particular? Forming soluble and stable protein structures with tightly packed cores is essential for stabilizing proteins and creating a rigid framework with well-defined binding sites. This requires the diversity of amino acids used in cells. A selection of different hydrophobic amino acids provides more options for building a tightly packed protein core and forming specialized pockets to enable a variety of enzymatic functions. On the other hand, proteins required on the surface, such as those in cell membranes, need straight and polar side chains like those of arginine and glutamic acid. The specific combination of these 20 amino acids allows for the precise structural and functional versatility necessary for life. A paper by Andrew J. Doig, 2016, noted, why the particular 20 amino acids were selected to be encoded by the genetic code remains a puzzle. They were selected to enable the formation of soluble structures with close-packed cores, allowing the presence of ordered binding pockets. The paper, Frozen, but no accident, why the 20 standard amino acids were selected, concluded remarkably, we find that there are excellent reasons for the selection of every amino acid. Rather than being a frozen accident, the set of amino acids selected appears to be near ideal. Another paper of Nature magazine, Extraordinarily Adaptive Properties of the Genetically Encoded Amino Acids, reported in 2015, we drew 100 million random sets of 20 amino acids from our library of 1913 structures and compared their coverage of 3 chemical properties, size, charge, and hydrophobicity, to the standard amino acid alphabet. We measured how often the random sets demonstrated better coverage of chemistry space in one or more, two or more, or all three properties. In doing so, we found that better sets were extremely rare. In fact, when examining all three properties simultaneously, we detected only 6 sets with better coverage out of the 100 million possibilities tested. That's quite striking. Out of 100 million different sets of 20 amino acids that they measured, only 6 are better able to explore chemistry space than the 20 amino acids that life uses. That suggests that life's set of amino acids is finely tuned to 1 part in 16 million. The problem here is that molecules and an arrangement of correctly selected variety of amino acids would bear no function until life began. Only a conscient intelligent agent with foresight and reasoning could select for specific distant purposeful goals. Random, unguided events produce only chaotic structures. Science is absolutely clueless about how the optimal set could have emerged prebiotically and without a guiding hand. The three main building blocks used in life, namely amino acids, ribose, the backbone of nucleotides, and phospholipids, are homochiral, that is, they possess the same handedness or chirality. Homochirality of biological molecules is a signature of life. The chirality or sense of handedness of the amino acid molecules is an important problem. Figure above shows 2 versions, one is left-handed, the other right-handed. Each contains exactly the same number of elements with the same types of chemical bonds, and yet they are the mirror image of each other. A molecule that is not superimposable on its mirror image is called chiral. The handedness of biological molecules such as amino acids or nucleotides plays a role in their functionality. In proteins, only left-handed amino acids can be incorporated. Cells, for instance, use complex molecular enzymes and proteins to synthesize amino acids only in left-handed form. The origin of homochirality remains a mystery, even after 175 years, since 1848, when Louis Pasteur observed the optical activity of tartaric acid crystals. Let us even grant for a moment that prebiotic processes like chiral catalysis or polarized light would have initiated a chiral bias. Modern cells enforce homochirality through at least 12 complex enzymatic systems. The transition from prebiotic asymmetry to today's highly regulated, enzyme-driven processes highlights a key leap, and remains a central problem in understanding how such a leap could have been crossed by unguided means. Furthermore, cells employ a sophisticated system to ensure only left-handed, L-form, amino acids are used in proteins. Aminoacyl tRNA synthetases perform error checking by hydrolyzing incorrectly attached D-amino acids, while D-amino acid oxidase and proteases degrade or recycle misformed or D-amino acids. Chaperones like HSP70 and GROEL/GROES further ensure proper protein folding, preventing the incorporation of D-amino acids that could disrupt cellular function. The transition from prebiotic production of left-handed amino acids to the exclusive use of L-amino acids in life is highly unlikely due to a vicious circle. The very proteins and enzymes needed to enforce homochirality, such as aminoacyl tRNA synthetases, D-amino acid oxidase, and chaperones, would themselves require L-amino acids to function. Without these error-checking and repair mechanisms, any prebiotic L-amino acid production would be quickly overwhelmed by racemization or contamination with D-amino acids. This interdependence creates a paradox, making the emergence of homochirality through gradual, unguided processes extraordinarily improbable. The formation of amide bonds without the assistance of enzymes poses a major challenge. Given an ocean full of small molecules of the types to be produced on pre-biological Earth with the types of processes postulated by the origin of life enthusiasts, the next question is about polymerization. There are many different problems confronted by any proposal. Polypeptides are polymers of amino acids. In other words, many amino acids are chained and linked together via dehydration reactions that form peptide bonds between them. Polymerization is a reaction in which water is a product. Thus it will only be favored in the absence of water. The presence of precursors in an ocean of water favors depolymerization of any molecules that might be formed. A peer-reviewed paper published in 2014 tried to answer the question of prebiotic polymerization on early Earth. Prebiotic amino acid polymerization faces challenges such as racemic mixtures, which hinder functional polymers, and the lack of plausible concentrating mechanisms, causing dilute and unstable conditions for polymer formation. These issues undermine the feasibility of abiotic pathways for generating biologically relevant polypeptides. Another problem is the fact that high activation energies are required for amide bond formation, beside the fact that direct peptide bond formation is slow unless high temperature or activating agents are used. Another problem is that there are 4 different ways to bond amino acids together by the side chains. If bonded to the wrong side chain, no deal. Proteins are linear polymers formed by linking the alpha-carboxyl group of one amino acid to the alpha-amino group of another amino acid. To form a chain, it is necessary that only bifunctional monomers react together, that is, molecules with 2 functional groups so they combine with 2 others. If a unifunctional monomer, with only one functional group, reacts with the end of the chain can grow no further at this end. If only a small fraction of unifunctional molecules were present, long polymers could not form. But all prebiotic simulation experiments produce at least 3 times more unifunctional molecules than bifunctional molecules. Polymers can adopt various chain structures: linear, branched, cross-linked, or networked. Proteins are linear homopolymers, while peptide polymers branched. There is no restriction of amino acids bond and form any of the different polymer structures. But only homopolymerization permits the formation of functional primary structures used in proteins. Obviously, there was no natural selection of the right formation on the prebiotic Earth. In the same sense as the sequence of alphabetic letters are required to write words, the right sequence of amino acids are required to make functional proteins. This is truly fascinating, in the same sense as the instructional complex blueprint specifies how to make each gear wheel of a watch, and ultimately, the watch itself, the genetic code instructs the right amino acid sequence to make functional proteins. If we consider that there were no molecular cell machines to make proteins, no evolution, no mutations or natural selection prior to functional cells and DNA replication, how could the right sequence of a minimal set of at least 1200 proteins required for life have been specified, if not by an intelligent agency? We only know of intelligence producing instructional information to make machines and factories for specific goals and purposes. Proteins, in order to become functional, must fold into very specific 3D shapes, which happens right when they come out of the ribosome, where they are synthesized. Specific protein shape and conformation depends on the interactions between its amino acid side chains. For a protein to function, it must fold into a resting state, which is a complex 3-dimensional structure. If a protein fails to fold into its functional structure, then it is not only without function but it can become toxic to the cell. As proteins fold, they test a variety of conformations before reaching their final form, which is unique and compact. Folded proteins are stabilized by thousands of non-covalent bonds between amino acids. A relatively small protein of only 100 amino acids can take some 10 to the power of 100 different configurations. If it tried these shapes at the rate of 100 billion a second, it would take longer than the age of the universe to find the correct one. Just how these molecules do the job in nanoseconds, nobody knows. Even if somehow a polypeptide chain in a functional sequence would emerge and fold into a form that later can be useful in the cell, that does not explain how to get to proteins which use not rarely several chains. Most proteins become only functional if they are joined into hollow enzymes, where various amino acid chains come together like lock and key. Some proteins complexes use over 50 chains or subunits to form a highly complex molecular protein machine. And each subunit must fit precisely to interact correctly with other subunits or protein chains. Ribosomes, for example, which are veritable macromolecular factories, are life essential, and found in all living cells. They are the factory which produce biological protein synthesis by translating the genetic code, and had to be fully developed when self-replication and life started. Each ribosome contains around 80 proteins which work together like a various machine parts in a joint venture. It exerts far tighter quality control than anyone ever suspected over its precious protein products. And discards error-laden proteins 10,000 times faster than it would normally release error-free proteins. But even if there would emerge a functional ribosome factory on the early Earth, by itself, it would be like a piston outside the engine block of an automobile. Many proteins bear only function once they are integrated in an assembly line, producing sophisticated molecular products used in life. To make proteins, and direct and insert them to the right place where they are needed, requires systematic cooperation. So the origin of this machinery cannot be explained through evolution. At least 25 unimaginably complex biosyntheses and production line-like manufacturing steps are required. Each step requires extremely complex molecular machines composed of numerous subunits and cofactors, which makes its origin an irreducible catch-22 problem. In order for evolution to work, this awe-inspiring robot-like working machinery and assembly line must be in place, fully operational. All it is left as alternative explanation to intelligent design, are random chemical reactions. Does that make sense? Now, let's suppose all this would assemble by a freaky random accident on early Earth. There would be no mechanism of transition from a prebiotic assembly to cell factory synthesis. The smallest bacterium is so much more like people than Stanley Miller's mixtures of chemicals, because it already has these system properties. So to go from a bacterium to people is less of a step than to go from a mixture of amino acids to that bacterium.
This video is based on the new trilogy called X-ray of Life, which represents the first truly integrated investigation of life's origin ever conducted, synthesizing insights from biochemistry, information theory, systems biology, and engineering principles. The results are devastating for those who held hope that advancing scientific knowledge would eventually yield a naturalistic explanation for life's emergence. Paradoxically, it is precisely our deepening scientific understanding that has revealed insurmountable barriers to unguided scenarios. This trilogy uniquely tackles the origin of life challenge with meticulous attention to 1. Comprehensive scope, covering every major topic from prebiotic chemistry to the functional integration of life. The work identifies over 2,000 unresolved questions. 2. Interdisciplinary integration, insights from biochemistry, systems biology, and engineering principles are interwoven to highlight the complexity of life's earliest stages. 3. Critical analysis, every mainstream hypothesis, from RNA world to metabolism first models, is examined with detailed critiques of their limitations. 4. Focus on simultaneous systems emergence, emphasis is placed on the difficulty of multiple interdependent systems emerging without external guidance, which is rarely discussed in mainstream origin of life texts. Volume 1 lays the groundwork by examining prebiotic chemistry and the formation of essential molecules. It addresses the challenges of synthesizing and assembling essential biomolecules under prebiotic conditions. It covers key prebiotic reactions, the origin of molecules like amino acids, nucleotides, and lipids, and the challenges of assembling them into functional systems. Volume 2 focuses on cellular emergence, detailing the transition from molecules to integrated systems. Examines the interdependent emergence and integration of metabolic, genetic, and structural systems required for cellular life. Examines the emergence of cellular systems, including metabolism, genetic replication, and compartmentalization. Volume 3 culminates with a focus on system integration, complexity, and the probabilistic challenges of minimal life. Analyzes the improbability of life's origin through chance, highlighting the coordination and complexity of molecular and population-level systems, explores the genetic code, protein folding, cellular homeostasis, and the probability of assembling minimal life forms. Discusses the full integration of early life systems, addressing signaling, regulation, and the emergence of complex networks in early prokaryotes. The link to buy the books in Kindle or paperback is in the description. If you enjoyed the video, please give a like, and subscribe to the channel. Thank you.